Welcome, everyone. Today, we are diving into the fascinating world of enzyme nomenclature and classification. If you have ever wondered how scientists across the globe communicate about the thousands of complex chemical reactions happening inside our bodies without getting completely confused, you are in the right place. We are going to learn the universal language of biochemistry. To understand why we need a naming system, let us start by imagining a massive, bustling factory with thousands of workers. But everyone is just called, "Hey you," or given a random nickname. It would be absolute chaos. Historically, this is exactly what happened in biochemistry. Scientists named enzymes whatever they wanted, often just adding the suffix "in," like pepsin or trypsin. While these names are catchy, they tell us absolutely nothing about what the enzyme actually does. As researchers discovered thousands of these microscopic workers, this random naming system became a complete nightmare for global communication and scientific research. Because of this growing chaos we just discussed, the scientific community had to take action. The International Union of Biochemistry and Molecular Biology, or IUBMB, stepped in to create a standardized system. They decided that enzyme nomenclature must be a formal, systematic method based strictly on the specific chemical reaction the enzyme catalyzes. Instead of random historical names, every enzyme is now assigned a systematic name. This name clearly states the substrate it works on, the exact type of reaction it performs, and it always ends with the suffix "ase." This ensures that anyone reading the name instantly knows the enzyme's job. Now that we know what the IUBMB system is, you might wonder why this strict naming system actually matters in the real world. Imagine a doctor trying to diagnose a patient with a failing liver. By looking for specific enzymes, like alanine aminotransferase in a blood test, the standardized name tells the doctor exactly which chemical process is failing. In pharmaceutical research, scientists designing drugs to block a specific bacterial enzyme must know its exact classification to build a molecule that fits it perfectly. Without this universal language, medical databases would be unsearchable, and global scientific collaboration would grind to a complete halt. This universal language we just explored relies heavily on categorization. To make sense of thousands of reactions, the Enzyme Commission divided all known enzymes into 6 primary classes. This classification is based entirely on the fundamental nature of the chemical transformation they manage. This means that enzymes doing similar types of chemical work are grouped together, regardless of what specific molecules they are working on. To help you remember these 6 major classes in order, biochemists often use the handy mnemonic OTHLIL. Let us break down exactly what each letter of this mnemonic stands for. Following our OTHLIL mnemonic, let us look at the first 3 classes. Class 1 represents oxidoreductases. These enzymes catalyze oxidation-reduction reactions, meaning their primary job is transferring electrons from one molecule to another. Next, Class 2 represents transferases. Just as the name implies, these enzymes move a specific functional group, like a phosphate or methyl group, from a donor molecule directly to an acceptor molecule. Then we have Class 3, the hydrolases. These are crucial for processes like digesting the food we eat. They break chemical bonds by forcing a water molecule into the structure, effectively splitting the target molecule apart. Moving on to the second half of our mnemonic, we encounter classes 4 through 6. Class 4 consists of lyases. These enzymes also break bonds, but unlike hydrolases, they do it without using water or oxidation, often forming a new double bond in the process. Class 5 is the isomerases. These are the reorganizers. They simply rearrange the existing atoms within a single molecule to create a different isomer, without adding or removing anything. Finally, Class 6 represents the ligases. Think of these as biological glue. They join two large molecules together, a heavy-duty process that usually requires an input of cellular energy from ATP. Now that we understand the 6 major classes, we can look at the mathematical format used to identify every single enzyme. This comes from the Enzyme Commission, which assigns a specific 4-digit code. The formula is EC followed by 4 variables, A, B, C, and D. The first variable A is the major class, from 1 to 6 that we just learned. If this number changes, the fundamental chemical reaction is completely different. The variable B is the subclass, telling us the exact bond or group acted upon. The variable C is the sub-subclass, providing finer details like specific electron acceptors. Finally, D is just a serial number, indicating the order in which the enzyme was discovered. Let us see how this works in practice. To see how powerful this formula is, let us decode a famous enzyme called hexokinase, which is given the code EC2711. We need to map these 4 digits to our classification rules. We start with the first digit, 2. This tells us immediately that its major class is transferases, so it moves a functional group. The second digit, 7, is the subclass for transferring phosphorus-containing groups, specifically a phosphate. The third digit, 1, tells us the acceptor is an alcohol, specifically a hydroxyl group. The final digit, 1, is just its historical serial number. Putting it all together, we instantly know hexokinase is a transferase that moves a phosphate group onto a hydroxyl group, which is the crucial first step in cellular respiration. Let us reinforce what we just decoded by looking at this visual breakdown of the EC number. At the top, you see the full code for hexokinase. Notice how each digit branches down into a specific layer of information. The first arrow points to the broad class, transferase. As we move to the right, the arrows point to increasingly specific chemical details. The subclass narrows it down to a phosphate transfer, and the sub-subclass pinpoints the exact docking station, the hydroxyl group. Finally, the serial number on the far right gives us the specific identity of the enzyme. This diagram perfectly illustrates how a simple 4-digit code funnels down from a broad category to a highly precise biochemical description. Now that we have decoded an existing enzyme, let us look at the chemical equation for a brand new reaction. Suppose you are a researcher who discovers an enzyme in a deep-sea bacterium. You observe that it takes a molecule of glutamate and a molecule of ammonia and uses the energy from breaking down ATP to join them together into a new molecule called glutamine. In this formula, glutamate and ammonia are our building blocks on the left. ATP is the energy currency being spent, which is why it turns into ADP and inorganic phosphate on the right. If we increase the amount of ATP and reactants, the enzyme will produce more glutamine. Our goal is to determine which major class this enzyme belongs to. Let us solve this classification problem step by step. First, we are given the chemical equation, and we need to find the enzyme's major class. We start by examining the fundamental chemical change. We see 2 separate molecules being combined into a single larger molecule. Next, we look at the energy dynamics. The breakdown of ATP clearly shows this fusion requires an input of cellular energy. We can rule out hydrolases because no water is breaking bonds, and we can rule out oxidoreductases because it is not a simple electron transfer. Because the enzyme acts as a biological glue driven by energy, it perfectly fits the definition of class 6. Therefore, this new enzyme is a ligase, and its EC number must start with a 6. We have covered a lot of ground today, so let us summarize the most important ideas. First, we learned that modern enzyme nomenclature completely replaces chaotic historical names with a highly organized logical system. Second, we saw how the 4-digit enzyme commission code acts as a precise map, guiding us from a broad reaction type down to specific molecular targets. Third, remembering the 6 major classes using the OTHLIL mnemonic gives you the foundational key to unlocking how every biological reaction is managed. You now have the tools to read and understand the universal language of biochemistry. Great job, and keep exploring the incredible microscopic machinery that keeps us alive.